Hello everyone, my name is Arlynn Ozara Nyohadinata.
And I'm Nadia and Timotius. We are from grade 8 junior high school and today we are going to talk about our research.
Our research topic is about the making of bioethanol from Chempedak straw. Why do we choose this topic? Because recently there has been news from CNN on March 25, 2020 about the scarcity of hand sanitizer. This is because due to COVID-19, the demand of hand sanitizer gets higher. Hand sanitizers are made of ethanol. The demand of ethanol will increase together with the demand of hand sanitizers. There are two types of ethanol. The first one is synthetic ethanol, or we can call it as ethanol, and the second one is bioethanol. What's the difference between these two? That bioethanol is more environmentally friendly. And what's the Chempedak for? So Chempedak is a fruit from East Kalimantan. It contains carbohydrate which can be used as the raw material for making bioethanol. One of the parts from Chempedak is the straw. Usually people tend to throw the straw since it doesn't have that much benefit and it became trash. But for our research, it'll be useful and it has two benefits. We can reduce the amount of trash, and the demand for raw material for making hand sanitizers can be provided locally.
For our research methodology, we cannot do the experiment. Why? Because the number of people who got infected by COVID-19 is the highest in East Kalimantan. In addition, the material needed to carry out this experiment are still incomplete at our school. Actually, we wanted to do the experiment in Mullah Warman University, but because of the pandemic, the university was closed. So we are going to use the secondary data to replace this experiment. The first objective of this research is to know the effect of the weight of the Saccharomyces cerevisiae toward the volume of bioethanol produced. And the second is to know the characteristic of bioethanol from Chempedak straw toward some bacteria. We also have some problem to be solved during our research, which are what is the effect of the weight of Saccharomyces cerevisiae to the volume of bioethanol produced, and how is the antimicrobial ability of bioethanol from Chempedak straw.
The process making of bioethanol. First, you prepare the Chempedak straw. Second, you clean and soak it with salt water or degumming for around 5 minutes. Third, is the day dry the process by putting the Chempedak straw onto the oven around 105 degrees Celsius. The oven method was more effective than the sun drying, and the ovening process was done to remove the water from the Chempedak. The fourth step is to crush the Chempedak until its texture becomes like flour. And the fifth step is by doing the hydrolysis process using HCl. The purpose of the process is to break down carbohydrates or starches in the form of long-chain saccharides into glucose or short-chain saccharides. Chempedak straw then stirred in water with HCl so that the HCl concentration becomes 0.6 molar. The process was done around 1.5 hours and the constant temperature is 90 degrees Celsius. The sixth step is the fermentation process using the Saccharomyces cerevisiae. Glucose that has been hydrolyzed is then mixed with a 0.05 grams of urea and 0.05% of NPK, then added with the Saccharomyces cerevisiae as much as 25, 50, 75, and 100 grams. Then leave it in a light tight room for 10 days. The seventh step is the filtration process, is to separate the solid from the liquid. And the last step is the distillation process, which is to separate the water and impurities. The distillate form is then known as the bioethanol.
And now we are going to talk about the antimicrobacteria testing. As the raw material for hand sanitizer, the bioethanol will be tested by MIC and MKC. MIC, minimal inhibitory concentration, is the lowest concentration of an antimicrobial. MKC is the smallest actual concentration that can kill 99.9% of bacteria carried out by growing bacterial test result on collagen media with treating method. The first step is to add 0.1 ml of bacteria in each tube. Then we incubate all the tube for 24 hours in 37 degrees Celsius. After that, we will observe the turbidity, tube number 1, 2, and 3, compared with tube number 4 and 5. Tube with the lowest clarity is the MIC, minimum inhibitory concentration. The next step is to trace every tube on the culture media, Na, and incubate for 24 hours in 37 degrees Celsius. Finally, the last step is culture media with the lowest growth of bacteria is the MKC, minimum killing concentration. So we are going to test out by putting solution for tube number 1 with 0.25 ml of bioethanol and 1 ml of DMSO. Tube number 2 made from 0.5 ml of bioethanol and 1 ml of DMSO. Tube number 3 made from 1 ml of bioethanol and 1 ml of DMSO. Tube 4, to be the positive control, the solution made from 1 gram of chloramphenicol and 1 ml of aqueous. Tube 5, negative control, the solution is 1 gram of DMSO in 1 ml of aqueous.
For the bioethanol analysis, we got this research data from Irfan, Ayuwanira Putri, Sri Ulina, Surbakti, and Baman Trisakti. The research topic is about the effect of the concentration Saccharomyces cerevisiae and the fermentation duration to the volume of bioethanol from the seed of Chempedak. We use this data because both of us observed Chempedak. But the difference is that our research observed the straw, while Irfan and friends observed the seed. The table shows the relation between the weight of Saccharomyces cerevisiae with the volume of bioethanol produced. The distillation result is in period, so to get the volume of pure bioethanol, the distillation volume must be multiplied by its purity percentage. The data shows a 3-day fermentation. From the table, when the weight of Saccharomyces cerevisiae increases by 3 grams, the volume of bioethanol increased by 15%. And when the weight increases by another 3 grams, the volume increased by 1.6%. So the more the weight of Saccharomyces cerevisiae, the more the bioethanol produced. However, the percentage volume increase is getting smaller. Why? There will be a maximum point between the increased volume of bioethanol and the weight of Saccharomyces cerevisiae. The more weight of Saccharomyces cerevisiae, it will also increase the acidity of the container and influence of potential hydrogen, or pH. The acidity of the container will influence the volume of bioethanol that's produced. In 1998, Saroso said that the best pH for the Saccharomyces cerevisiae to produce the bioethanol is 4 to 4.5 pH. If it's more or less, the volume will decrease.
Now we will talk about the analysis antimicrobiethanol. We got this data from the result of a research conducted by Rina Saputri, Ali Rahman Hakim, Dahlia Syarani, Fatia Lisan from Salim Mulia University, Banjarmasin. Here is the data of the MIC observation result. The positive symbols means that the bioethanol has the ability to restrict the growth of the bacteria, and the negative symbols means that the bioethanol don't have the ability to restrict the growth of the bacteria. Next is about the MKC observation result. The positive symbols means that the bioethanol has the ability to kill the bacteria, while the negative means that the bioethanol don't have the ability to kill bacteria. The data appeared on table 3 reveals that the bioethanol with 50% above can kill bacteria E. coli and S. aureus. Both tables show that when the concentration of bioethanol is 50%, it can kill bacteria E. coli and S. aureus. In the same concentration, bioethanol also has the ability to restrict the growth of the bacteria. So if there are any industry wants to use this bioethanol as the raw material for hand sanitizer, bioethanol with 50% concentration is enough.
Finally, the conclusion is the observation research shown on Table 1. For fermentation duration in 3 days, the more weight of Saccharomyces cerevisiae, then the more bioethanol produced. In this case, when the weight of Saccharomyces cerevisiae is 9 grams, then the pure bioethanol produced is 2.624 milliliters. Observation results appear on Table 2 and 3 that bioethanol with concentration 50% or above can restrict and kill bacteria E. coli and S. aureus. As a conclusion, the bioethanol from Chempedak straw is able to be the material for hand sanitizer. For our future work, further research and analysis about the effect of the fermentation process to the volume of bioethanol produced. It is recommended further study and analysis for the percentage of pure bioethanol in the distillate.
I'm Nadia.
And I'm Arlynn. Thank you for listening.